Good morning, student. In last lecture, we have studied about mitosis and meiosis. Today, we'll see next point, that is nucleic acid type, structure, and function. We know there are two types of nucleic acid, that are deoxyribose nucleic acid and ribose nucleic acid. So, first we'll study about deoxyribose nucleic acid. Structure of DNA. We'll see now. Deoxyribose nucleic acid, DNA. It is a component of a chromatin, which is the largest and made up of a complex biomolecule present mainly in nucleus but also found in cell organelles like chloroplast and mitochondria. DNA was first described by Watson, Crick and Wilkins in 1953. They were awarded Nobel Prize in 1962. However, the first X-ray diffraction photograph of DNA was taken by Rosalind Franklin in 1953, suggesting double helical structure of DNA. This DNA has nucleotide, and nucleotide is a compound formed by combination of nucleoside and phosphate group. Nucleotide building blocks of nucleic acid, and nucleotide have three characteristic components, that are nitrogenous bases, then pentose sugar and phosphate group. Then first we'll see nitrogen bases present in DNA. In DNA, purine and pyrimidines ratio is always equal, that is 1:1. So there are two nitrogen bases are present, purines and pyrimidines. Then again, purines having two nitrogen bases, adenine and guanine, and pyrimidines have thymine and cytosine. In short form, we are calling adenine as A, guanine as J, thymine is T, and cytosine as C. So there are four nitrogen bases, A, G, T, C. Then the structure of DNA, Watson and Crick model. So the DNA consists of two strands are coiled on each other, and both the strands coiled on a common hypothetical uh, axis. So this it forms a double helix coiling twice. The double twisting of the two strands result into major groove and minor groove. It consists of two helical chain wound around the same axis to form a right-handed double helix. The hydrophilic backbones of alternating deoxyribose and phosphate group are on the outside of double helix. Purine and pyrimidine bases of both strands are stacked inside the double helix. The hydrophobic planar ring structure very close together and perpendicular to the long axis. Then of the two strands, one is placed in ascending direction while the other is in descending direction. Hence DNA molecule is polarized and the two strands are antiparallel. Here we can see two strands. One is in ascending direction and other one is in a descending direction. These are antiparallel. Each strand of DNA is made up of several nucleotide linked in the chain to form polynucleotide. Each nucleotide is made up of molecule of deoxyribose sugar, which is pentose sugar, then phosphate group in the form of phosphoric acid and nitrogen base. Each phosphate group is joined to third uh, carbon atom. Here we can see each phosphate group is joined to third carbon atom of one deoxyribose molecule and fifth carbon atom of successive deoxyribose molecule of the same strand by phosphodiester bond. Nucleotide without phosphate group is called as nucleosides. Then for their outstanding work of Watson, Crick and Wilkinson, uh, and discovering the double helical structure of DNA, the Watson and Crick shared the shared the Nobel Prize in 1962 for physiology and medicine with Wilkins, uh, Sidley, and Franklin, whose working greatly contributed to this key discovery. Then we'll see form of DNA. In both DNA strands, the nitrogen bases are attached to first carbon atom of sugar molecule and uh, that forming glycosidic bond, and at right angle to the sugar phosphate backbone. The nitrogen bases are of two types, purine and pyrimidine, that we have seen. Then now we'll see form of DNA. There are two form of DNA, aDNA and bDNA. Through analysis of the early DNA diffraction study showed that DNA can exist in different form depending on the condition. At high humidity, DNA exists in B form. This form deduced by Watson and Crick. When humidity is relatively low, DNA is found in the A form, and another form of DNA as Z form has also been identified. So first we'll see aDNA and bDNA. Both aDNA and bDNA are right-handed double helix, um, 10.9 and 10.0 base pair per 360 degree turn of the helix respectively. Then the bases are inclined away from the perpendicular from the helix axis by 13 degree and 2 degree in aDNA and bDNA respectively. The double helix of aDNA is short and wide with narrow and very deep major groove and wide shallow minor groove. The bDNA double helix is thinner and taller for the same number of the base pairs than aDNA, with a wide major groove and narrow minor groove, and the depth of the both the groove is the same. These are the two form of both DNA, aDNA and bDNA. Then next is zDNA. zDNA is a left-handed DNA helix with zigzag sugar phosphate backbone. It has 10 it has 12 base pair per complete helical turn. The bases are inclined away from the perpendicular with the helix axis by 8.8 degree. The double helix of zDNA is thin and elongate with a deep minor groove. The major groove is pushed to the surface of the helix so that it is not really evident as a distinct groove. In zDNA, the negatively charged phosphate group are brought closer together than bDNA, causing the phosphate group to repel each other. This is responsible for making zDNA less stable than bDNA. Then zDNA is rich in cytosine and guanine base pair. Then we'll see function of DNA. DNA contains genetic information in a coded form. DNA transfer genetic information from one cell to another cell by the process of replication. DNA decides and regulates which type of protein and protein enzyme are to be synthesized in the cell. DNA can cause mutation by sudden changes in nucleotide sequence. Then next is ribonucleic acid. Uh, it is also called as RNA. It is present in nucleus as well as in cytoplasm. It is mostly single stranded, but sometimes it is found to be double stranded. Single stranded is made up of alternate molecule of pentose sugar phosphate linked together. In RNA, nitrogen bases are similar to purines like adenine and guanine, and to pyrimidines like cytosine and uracil. Here, instead of thymine, uracil is present. RNA are of two types: genetic RNA and non-genetic RNA. Genetic RNA. So genetic RNA plays genetic role. It replicates and transfer genetic information to next generation. The replication of genetic RNA takes place by reverse transcription through about by enzyme reverse transcriptase. The RNA of the virus is released into cytoplasm of the host. The enzyme reverse transcriptase synthesize complementary DNA using RNA as a template. This single-stranded DNA synthesizes its complementary anti-parallel strand, thus forming a double-stranded DNA is called as provirus. This uses the synthetic machinery of the host cell to produce several copies of RNA and protein molecule used in the formation of viral protein code. Then non-genetic RNA. Non-genetic RNA does not replicate but it performs cellular function like protein synthesis. It is differentiated into three types: messenger RNA, then ribosomal RNA, transfer RNA. So there are three types of RNA: mRNA, rRNA, and tRNA. So messenger, first we'll see messenger RNA. Messenger RNA is a single stranded RNA molecule that is complementary to one of the DNA strand of a gene. The mRNA is an RNA version of the gene that leaves the cell nucleus and moves to cytoplasm where proteins are made. During protein synthesis, an organelle called ribosome move along the RNA, read its base sequence, and uses genetic code to translate each three base triplet or codon into its corresponding amino acid. Then function of mRNA. Messenger RNA or mRNA plays vital role in human biology, specifically in process known as protein synthesis. It is single-stranded molecule that carries genetic code from DNA in cell nucleus to ribosome, the cell's protein-making machinery. So it is important for protein synthesis. Then ribosomal RNA. Ribosomal RNA is longest RNA, near about 80% of total RNA. It is synthesized in nucleus by DNA and process is catalyzed by RNA polymerase third, and its final organization takes place in the cytoplasm. So then structure of rRNA, the structure of rRNA able to drastically change to affect tRNA binding to the ribosome during translation of other mRNA in 60s RNA. Uh, this is thought to occur when certain nucleotide in the RNA rRNA appear to alternate base pairing between one nucleotide or another, forming a switch that alters the rRNA's conformation. This process is able to affect the structure of the LSU and SSU, suggesting that this conformational switch in the rRNA structure affect the entire ribosome in its ability to match a codon with its anticodon and tRNA selection as well as decode mRNA. Function of rRNA. rRNA provide proper binding site for mRNA on the ribosome to orient mRNA in such a way that its nitrogen based triplets or codons are properly knit to release tRNA from mRNA after transfer of activated amino acid to uh, mRNA uh, chain. Then transfer RNA or tRNA. Transfer RNA is shorted RNA found in cytoplasm. It is synthesized in nucleic acid or nucleus. It is synthesized in nucleus and it's constitute about 15 to 20% of the total RNA. Here we can see one of the model of tRNA cloverleaf model. According to the cloverleaf model, different region can be recognized in the tRNA molecule. The amino acid arm, acceptor arm, then anticodon arm is situated opposite to amino acid arm and consists of stem and a loop known as anticodon loop. Then DHU stem and loop has an enzyme, then recognition site for binding to the tRNA to its specific amino acid. Then the variable arm consists of 4 to 21 nucleotide. Then function of tRNA. The anticodon of tRNA recognize the codon of mRNA as it is complementary to latter. Hence, it is also called as recognition site. As per the codon, the tRNA having complementary anticodon picks a specific activated amino acid from cytoplasm and transfers it to polypeptide chain that is being formed on uh, mRNA ribosome complex. Thus, tRNA helps to arrange different amino acid in proper sequence to form specific protein. So, uh, in this way, we have seen two types of nucleic acid, DNA and RNA. We'll see comparison between DNA, RNA. DNA. First, we'll see DNA. DNA is mainly in the nucleus, and RNA is present in nucleus as well and cytoplasm. DNA is double stranded, RNA is single stranded, and DNA pentose sugar is deoxyribose, and RNA pentose sugar is ribose. In DNA, thymine is one of the nitrogen base, and RNA in RNA thymine is replaced by uracil. In DNA, there presence of purine and pyrimidine equality, and RNA uh, does not shows purine and pyrimidine's equality. DNA is longer as it shows thousands of nucleotides. RNA is comparatively shorter as it shows few hundreds of nucleotides. DNA performs genetic role in all organisms and viruses. RNA plays cellular role in all organisms and viruses and genetic role in some viruses. DNA is functionally of only one type, and RNA is functionally of three types, viz mRNA, rRNA and tRNA. So in this way we have seen two types of nucleic acid, DNA and RNA. In this way we have seen this topic, that is cell biology, and in next lecture we have study next topic, that is cytogenetics and content of this topic. Thank you.